Okay, so let's go ahead and take a look at the next part here. And here we're going to look at a little bit of the terminology in relation to the DNA and then how the DNA affects the cell or how an organism looks. So the terms genotype and phenotype are what we're referencing here. So genotype is essentially the genetic makeup of an organism. It's basically what their genes are, what their genes look like, essentially, for lack of a better description there. We're going to talk about that in a little more detail next, but essentially what the DNA looks like, what the gene sequences are. The term phenotype is used to describe what those genes essentially do or how they become some characteristic of the cell. So, for example, you can have a sequence of DNA that essentially is a gene, okay? And we have information in the gene that describes how to make a protein. And this is an example I gave earlier, by the way. And it's because this is an easy example, okay? And then the protein makes a pigment, and the pigment leads to a color, okay? All of this here in the DNA, the genetic material, between here and here, the DNA and the gene. And just to give you a heads up, we're going to describe this here in a second, but just to give you an idea of what we're talking about here, this would look like this. It's a sequence, something like, not exactly, anyway, something like this. It's a sequence of random nucleotides, all of which are made up of only 4 letters, A, G, C, and T. And it's the exact way that these are described, in the exact order and sequence of them, that make up different genes in the sequence. So they're basically, it's like a language, basically. But it's a language that only is made up of 4 letters. And anyway, the language essentially spells out, the language rather is spelled out in the form of a gene, but made up of DNA. Okay, so that's the genotype. So this port right here, we can describe this as the genotype. So what do the genes look like? What's the genetic sequence? What are the 4 letters, A, T, C, and G? What do they look like? The way that ultimately manifests then is it manifests as a pigment, which creates a color. So on this end, we end up with what's called the phenotype, which is what it looks like. So this gene makes a pigment that makes it have a color. And we can say, for example, it's a red color. So the phenotype is a red color. Okay, so that's the difference between the two. Now, this is an easy example. Okay, they're not all that simple. Not every gene codes for a basic trait, so keep that in mind. But that's kind of the difference there. Okay, this next part we want to look at the DNA structure and function. So we're going to talk about the function and what's happening with DNA. And so to really be able to do that, you got to start with what it looks like. So structure of DNA exists as what's called a double helix. And there's a number of ways to draw it. Here's kind of a conventional way. And what you essentially have is what can be thought of as like a ladder, 2 rungs or 2 sides of the ladder, and then the rungs here in the middle. So this represents kind of the base of the molecule, and then it's actually twisted in a helical pattern like this. It's almost like a spiral staircase is a kind of a traditional way that they describe it. So it's kind of like a spiral staircase where you've got the outer parts here and then the inner parts, which are more like the steps of the rungs. This over here is more of like a realistic model. So it's a space filling model that's trying to show what it would actually look like from a molecular form, which is kind of confusing, and that's one of the reasons why it's not usually shown like that, because it's more difficult to really visualize. So that's more realistic. This is more of a better visualization. And then over here, what they've done is they just flattened it out to take out the twisting portion or that aspect of it, and just flatten it out so you can kind of see it better and what's really happening. So the twisting and the helical pattern does make a difference, but not for what we're going to talk about here today. So here we can see it in a flat pattern. And what we have is essentially 4 different types of nucleotides. And let's go ahead and let's skip to those here. So I was going to have you look at these, and I'm going to go to my paint here in just a second. But we essentially have 4 nucleotides, and they're described as adenine, guanine, thymine, and cytosine. Now, these 2 are of a class of molecules called pyrimidines, and these 2 here are from a class of molecules called purines. I don't really want you to know that much detail about all this, so I'm going to give you more detail than what I want you to know. Okay, but the reason I say that is because each group is chemically distinct, and what that does is it makes them react and interact in a unique kind of way. And so it turns out, through all this complex biochemistry, that adenine, a type of purine, really can only interact in a certain way with the pyrimidine thymine. So adenine and thymine, shown down here, A and T, they have a unique chemistry together. And that has to do with the fact that these purines and pyrimidines tend to react anyway, and that it just so happens that these 2 react in a very compatible way. The same is true of guanine and cytosine. Once again, a purine and a pyrimidine. So they have a compatible chemistry. So it turns out then that these 4 molecules always behave in a way that where adenine is found on one side of the molecule, you'll have a thymine on the other side. And the same is true of guanine and cytosine. Where guanine exists on one side of the DNA molecule, a cytosine exists on the other. So first of all, those are your 4 DNA nucleotides, and this is the complementary base pairing that goes along with that. And in fact, I've got a question on that right here, and this will be in the study guide also. What are the complementary base pairing rules? And we'll go back to those other questions here in a second. Now, going back to look at this here, what we can see then that on one side of a molecule, anywhere we have an A, T, C, or G, we have the complement on the other side. So here we have a G, here we have a C, here we have a T, A, etc., etc. So this is what's referred to as complementary base pairing. And it's one of those weird things about nature that it's hard to say exactly why it's that way. There are some reasons. We will talk about why complementarity is important, but it could have been drawn up anyway, and it's kind of an odd thing that happens. And so don't try to necessarily understand it, because it is what it is, and there's maybe not necessarily a good reason. It could just be that's the way it worked out. But what we see here is a molecule made up of 4 letters, 4 nucleotides, 4 chemicals. We can refer to them as letters. And actually, I'm going to go back to my analogy and fill this in here in just a second. But we can actually think of them as letters, and the way it works is that the combination and the sequence of these actually spells out information. Okay, so we can see that they're kind of arranged in this certain pattern. So anywhere we have one, we have the other, complementarity. Now, let's take another look here. There's actually 2 different types of bonds, chemical bonds, that make up the molecule. So one type of bond is called a covalent phosphate bond. And what you want to know about covalent bonds is that they're relatively strong, and that makes wherever they're found makes that part of the molecule a little sturdier. So we find these bonds on the outer, what are called what's actually referred to as the backbone of the DNA molecule, the phosphodiester backbone. So you have these nucleotides here. The nucleotide, by the way, is made up of this chemical group here, the adenine, guanine, cytosine, and thymine, but then it also has a sugar and then a phosphate to it. And so it's really kind of a 3-part molecule. This is called the nucleotide base, this is called the sugar, and this is called the phosphate. So base, sugar, phosphate. And each one of these has those 3 parts to it here. And what we see is that this phosphate group here actually forms the bond. So we call it a covalent phosphate bond. And each nucleotide connects to the next via a phosphate bond that occurs between them. Okay. Now it's not really like a little stick here. It's actually a chemical bond, like an invisible force, but they draw it with this little kind of like stick configuration, so you can kind of see how that would work. So that's really where it would look more like this. Really, these are all just kind of attracted to each other like magnets. So phosphate bonds are on the outside. So these occur on each side of the molecule. That makes the outside of the molecule relatively sturdy. There's another type of bond called hydrogen bonds, and they occur down the middle. And hydrogen bonds make this part of the molecule a little less sturdy. But hydrogen bonds are kind of like Velcro. If you think about Velcro, a little tiny piece of it doesn't hold very much, but lots and lots and lots of Velcro can actually hold quite a bit. So the idea is that each hydrogen bond between each of these two nucleotides is kind of weak, but when you add them all up in the entirety of the molecule, it's actually pretty stable. So each one of these is like a little piece of Velcro, and each one can be easily removed, but together they're all really strong. And what that does is it actually allows the DNA molecule to be split open right down the middle. And see if we can find a picture of that just real quick here. We would refer to that as DNA unzipping. Yeah, so DNA can do this here, where these essentially separate like that. So here they're showing the helix, here they're showing the bonds connected. And what can happen is essentially the two molecules will separate. And that's done because these hydrogen bonds are relatively weak. So each one can actually be broken and the two strands can separate. But because these are stronger, they tend to stay intact. They can break, they're not permanent, but they're definitely more sturdy. So that's the DNA molecule there. Now, one of the questions on here, why is complementary base pairing significant to DNA replication? And really the question should be, why is complementary base pairing significant? It's significant in one big way because it allows for accurate DNA replication. So I'm going to kind of put this in a couple of ways here. So first of all, let me back up here just a second. When we talk about DNA replication, just real quick, I want to just point out a few things here. So we're just going to mention DNA replication real briefly. I don't try to get into too much detail on this topic. It's relatively complex when you get into the details. But there's just a few basic things that I think are important to recognize. So when we talk about replication of the DNA, all we're really talking about is making a copy of the DNA. So when an organism wants to replicate itself, it needs to make a duplicate of everything. And if you remember from the lab, we discussed binary fission in the topic of bacterial population growth, okay? So the idea is that when an organism like a bacteria is going to replicate itself, it has to replicate the DNA. So we have a parent cell, and a parent cell is going to split apart and become two daughter cells. And those are just kind of random terms we use to describe this. Parent cell is the starting cell. Daughter cells are what are created. So when an organism does this, they need to duplicate the DNA. So what essentially is going to happen is it's going to take this DNA and it's going to make a copy of it. So we're going to now have two pieces. But the way it actually works is that the DNA is going to be first split down the middle, right down the middle, and it's going to be separated like this. That's the unzipping. Now, there's a lot of details that I'm omitting as to exactly how that works, so probably better that way. Okay, but what's actually going to happen then is when the DNA is split, what's going to come along is an enzyme that is going to actually replicate the DNA. And it's going to make a new strand. So let me just kind of show you here what happened. It's going to come along, and it's going to take, and it's going to build a new strand of DNA on each of the two strands that split apart. So we're going to see a new strand form here. I'm showing the new strand portion in red, and then the old strand in black. So each new strand is going to be replicated. And the way this works is that it uses complementary base pairing to make an exact sequence. So for example, let's just actually go back here just real quick and show that this works here. If we have a sequence of A, T, get this right here, ATCG, we would see on the other side a match. You guys get the idea here. I'm not lining these up perfectly. We would have a match of T, A, G, C. Sorry, that was painful. I know. But that's the idea. So there's a match there on each side. Okay. Now, when these split, carry this over here, we're still going to see that here, right? Because that's the same part of the DNA. So this part here is split, and that is what we're showing here in black. And now what we would see is that if we went to replicate this, we would end up with the same sequence over here, because the complementary base pairing, when it creates the new strand, it's going to use the same base pairing rules to make this strand over here. So this one's going to say T, A, G, C. Okay, so I'll spare you typing that in and trying to align it. So the idea is that we can essentially split the two molecule, there's the DNA molecule apart into two, and then use each of the base strands to make a new full strand and complete that complement. Okay, and so when you look at the daughter cells, then what would actually happen is that their DNA would actually be partly from the original parent and partly newly synthesized. And this is actually what's referred to as semi-conservative replication. And the idea is part of the DNA is conserved from the parent cell, part of it is newly synthesized. And interestingly, what that actually implies is that as a parent organism, you actually physically pass down your own DNA molecules to your offspring. And so if damage occurs to the DNA, that can actually physically be passed down to the next generation. So anyway, in a nutshell, that's why complementary base pairing is significant to DNA replication. You may have to look at that again and apologize for that, but that's the idea behind that. This last part, I want you to skip that, and I need to take that out. We'll look at that when we get to chapter 10. So just go ahead and skip that for now. When we look at, so that's in a nutshell how DNA structure relates to the replication of it. Okay, now let's talk about how the structure of the DNA creates endless variation, and let's talk a little bit more about the DNA. Let's just go back here real quick, and let's talk about the analogy I started using earlier. So I said the genome of an organism is like a book. I said the chromosomes are like the chapters, and then I said the genes are like the paragraphs / sentences. The last part really is the nucleotides. These are equivalent to letters. So these are essentially the individual units of the genetic language. And the idea is that just like a language, the exact sequence and the combination of these letters gives us different meaning. So if I put an obvious sentence on here, I don't have to say anything other than put those, just stop and think about this for a second. What I've done is I've put 9 letters in a certain pattern in front of you. That's all I've done. I just pulled out 9 letters and I put them in a very specific pattern, and you can actually then even visualize what that means. The same is kind of true for DNA. The difference is, instead of having 26 letters, it has only 4 letters, A, T, G, and C. But it basically does the same thing, which is use those letters to create really a language which translates into a meaning in the cell. It's actually pretty incredible how it works, really. And really, it's a language of 4 letters. Okay, so how does that work? And let's just talk about something else real quick. All organisms have only 4 letters. So everything from bacteria all the way up to humans is encoded by the exact same 4 letters. Same language, same letters, same everything. It would be like every living organism speaking English, except they have only 4 letters to the language. So everything kind of speaks the same language from a genetic standpoint, and that's actually why this here is called the central dogma of biology. It's called that because, as far as anyone can tell, all living organisms follow it, and there are no exceptions, like literally none. No one's found any yet. So it appears that every living thing does the exact same thing when it comes to this process, which is not necessarily something you'd expect. You might think that it would be different and that there'd be different ways of how this works, but it turns out it's basically identical in everything, which is kind of interesting. So how do you get so much variation out of 4 letters? Because you're encoding from everything to a bacteria to a human being with 4 letters. So how is that possible? It's possible because of the number of different ways that you can combine those 4 letters. One of the things about the English language is we have 26 letters, but we generally only combine them in up to about 8 ways. And I'm averaging here. An average word is actually probably about 6 letters. So we would essentially have 26 letters, and we could arrange them in an average of 6 ways. Okay, so with 26 letters, we can get, this is about 308 million different possibilities. So that's quite a bit. We have a lot of letters, and we have a fewer number of ways to combine those, or an average of, say, 6, give us about 308 million. So the idea is we have a big pool of letters, and with a smaller number of ways to arrange them, you get quite a bit of variation. Of course, we know we can have letter or words bigger than 6 letters, so we get even more possibilities. Really endless possibilities. To clarify, you have endless ways of describing and using these letters to make descriptions of the world. And with 26 letters, you have endless possibilities. So how does that work with only 4? 4 doesn't seem like enough. And the reason is because the way the DNA sequences are arranged actually occurs vertically on the DNA strand, going up and down like this. So we don't encode information horizontally, we encode it vertically. And typically the number of sequences like this that make up a basic gene are anywhere between 1,000 and almost 40,000. Okay, so in other words, you have typically at least 1,000 sequences, letters of A, T, G, C, G, G, C, C. There's no actual rules to how they can line up. So on a DNA strand, you can just see any number of possibilities and any possible combination here. Okay, and these sequences will go on for at least 1,000 positions, and some of the bigger genes go all the way up to almost 40,000. So the number of sequences per gene is fairly large. So when we do the math on this, what we see is we have 4 letters instead of 26, but even with a small gene, and we're not even using the average here, we're using a very small gene of, say, 1,000, we're basically taking 4 to the 1,000th power. Now, if you try and put that into your calculator, it's basically going to tell you that it doesn't work. It's going to tell you that error, computation error, something like that. It's basically bordering on infinite. It's not technically infinite. There's a number to it, but it is bordering on infinite. It is an incredibly huge number. And when you look at how exponents work, a small number to a large exponent of even a relatively large exponent like 1,000 is just a relatively huge number. Now, and that's a small gene. If you were to take 4 to the, let's say, 30,000 of, say, a relatively large gene, that number would just be incredibly huge. So, in other words, what we're talking about is how many different ways you can rearrange these to make new combinations. So if I take 6 letters or 6 units, 6-letter word, how many different 6-letter words can I create? Now, obviously, with the English language, we have rules, and we can't just take any 6 letters and put them together. So the same is actually somewhat true of DNA. So there's rules in both. That does limit the possibilities, just to clarify. It's not like this is the actual number of possibilities. But the truth is that there's so many different ways to rearrange them that there's really no limitation. And remember that we said a lot of organisms share DNA, so it's the way that they use the genes that makes even more variation. So we have really all this variation of life in reference to 4 basic letters. When you break it down, it's really almost hard to believe. But that is the way it actually works. That's how it's believable, because the number of possible ways that they combine these is infinite. So when you're looking at a DNA molecule, what you'd essentially be seeing is a great big strand of this that goes in one direction like this, and I'm not going to try and make these complementary, but then you'd have all the complementary DNA molecules on the other side like this. And these two strands would just go on and on and on and on and on and on and on. And at a molecular level, they're just almost infinite. So tons and tons of information. So when we look then at how we get so much information, it comes down to the numbers. There's no number or limitations really that these sequences can be arranged in on each side of the strand, and this gives us infinite possibilities. And so let me just go back real quick to clarify visually here. When we look at how a gene is encoded, how DNA becomes protein, information is actually encoded up and down the strand like this. I'm simplifying here, I'm not giving enough information, but the information from a gene to make a protein comes from a certain segment on just one side of a DNA molecule. So the information will be encoded going up and down the strand like this. Now I'm showing it vertically here just to go back. We're looking at the strand like this here. I was showing it horizontally, excuse me. Looking at it vertically, we'd be looking at the information being up and down one side like this. So interestingly, the DNA molecule has two sides to it, but the information for a gene only comes from one side of it. Okay, so that's where the information comes from. The other side is complementary and allows for the DNA to be replicated, and really each side only preserves one portion for information. Now that, as I say that, I will admit that that's not true entirely. I'm simplifying that, and new research may show that that's completely inaccurate. So that's not really the entire truth. I'm kind of trying to keep it without going into all the exceptions. But generally speaking, information is encoded on one side. And so we see that when you can add up all these different letters to make different possibilities, you have infinite ways to arrange these. Therefore, you have infinite gene possibilities. Therefore, you have different infinite protein possibilities. Therefore, infinite characteristics in the cell. So that's how that works. And I hope that makes sense. I know it's confusing, but that's hopefully gives you an idea of how we come up with all that. The numbers, and then the ways that you get infinite possibilities from those. The next part here, we're going to talk about RNA. So there's DNA and there's RNA, and they're very, very similar molecules. They're almost indistinguishable, except the difference is that they have a slightly different chemical group in one position. So here is a RNA nucleotide, and this goes back. We have the RNA. This is called the nitrogenous base. So this is like a purine singular ring. So just go back and clarify here. This is a DNA molecule. This is the ring. Oh, this is a pyrimidine. Excuse me, I misspoke there. This is the ring here, the pyrimidine ring. Here we see this is the whole nucleotide here, the ring, the sugar, and the phosphate. I should have a better picture of that. I apologize. Here's the three parts of it right there. RNA, three parts again: ring, sugar, phosphate. Here's DNA. Here's RNA, ring, sugar, phosphate. These would be pyrimidines. They're exactly the same in every way, except right here in this little spot on the sugar, there's an OH on an RNA and an H on the DNA. Doesn't seem like a big difference, but it changes the chemistry of the two molecules, and it creates a different set of properties. And that's definitely simplifying it. But that's really what you need to know. The naming, random biology jargon, as you'd expect. DNA stands for deoxyribonucleic acid, or individually a deoxyribonucleotide. Let's see if I can spell out. So you would refer to it as deoxyribonucleic acid. That's kind of almost like a plural sense. Here they're describing it in a singular sense, a deoxyribonucleotide singular. So deoxyribonucleic acid, RNA, is referred to as ribonucleic acid. And really, if you look, it's the same thing minus the deoxy. So deoxyribonucleic and then ribonucleic. And if you look, deoxyribonucleic has one less oxygen. Isn't that cute? So that's how they came up with that name. Deoxyribo and ribonucleotide. And that's why everybody hates biology. So that's the terminology. The difference partly lies there. There's another difference, and really a more practical difference, in that RNA is always going to be in a singular strand form. So part of the way that the chemistry changes is that it doesn't form the same kind of bonds anymore, and it actually forms a singular, or arranges in a singular strand. So we have a singular strand as opposed to a double strand. Similar, just half the equation, so to speak. So it doesn't actually, usually, doesn't, in this form here we're showing it anyway, not being necessarily paired with itself. It doesn't necessarily have the same way of complementary base pairing with DNA like DNA does. Another weird difference is that in RNA there is no thymine equivalent, and so there is actually a different molecule to replace thymine called uracil. And this is just one of those little weird quirks about life that this is just the way scientists observe it in the cell, and it's hard to say exactly why that is. So go back here just real quick. DNA consists of adenine, thymine, guanine, and cytosine. RNA consists of mostly the same things. Oh, this isn't going to work all that well, is it? You have an adenine RNA. Skip that one real quick. You have a guanine RNA, and you have a cytosine RNA. What you don't have is a thymine. And so instead of a thymine, you have a different molecule called a uracil. Why exactly that is, I really don't know. But for whatever reason, Mother Nature decided to replace thymine with uracil. So when we look at how the letters then are arranged, when we're talking about DNA letters, we can use the letters A, T, G, and C. However, when we talk about RNA letters, we have to use the letters A, U, G, and C. And so each of these designates the specific base pair. Now, when it comes to complementary base pairing, RNA will complementary base pair with DNA and vice versa, okay? And so in this case, when it comes to RNA base pairing with DNA, it turns out then that adenine is complementary to uracil. So anywhere you would see DNA and RNA interact, you would see the uracil complement with the adenine. So you wouldn't see the thymine. So it just really just replaces the thymine with the uracil. Going back to this, I don't have that on there. Never mind. Thought I had that on there. Thought I had the uracil. Here's showing the uracil nitrogen base here. So anyway, that's another difference with RNA. That's a little more practical because we'll be talking about how RNA is used here in a bit. Next here, we're going to talk about RNA's role in the cell. RNA does a lot, and so this is definitely a very simplistic view of the roles. This is a very traditional view of the roles. So this is not any of the new cutting edge stuff. In fact, I made a note about this when I put this together. If you're interested in that, then you could go look that up and learn more about it, because there's certainly a lot of really cool stuff to know there, just kind of outside the scope of this book, this chapter, or this class rather. So here's some information about that if you're interested. We're looking at the traditional roles. Some of the three of the most traditional roles are in the form of what's called messenger RNA, transfer RNA, and ribosomal RNA. Really, all of these are involved in the making of a protein, and we're going to talk about that in the next part here, 9.2, applications of the code. So when we get into that, we'll describe how these all kind of come together. But if you remember, ribosomes are what make protein. Think all the way back to chapters 4 and 5. We said ribosome synthesized protein, We didn't really say how they did it or even why, or just said that they do it. So here we're going to talk about kind of how that works in more detail. One of the things that happens is ribosomal RNA plays a big role in forming the ribosomes. So they're kind of like a big part of the building material for the ribosomes. So that's kind of their role, is that they basically make up the ribosomes. And then these molecules here interact with the ribosomes in a way that converts the building blocks of proteins into the actual proteins themselves. So think back to chapters 7 and 8. We talked about metabolism and growth, and I showed how amino acids are the building blocks for protein. So here we're going to talk about how these actually take those amino acids and convert them into protein chains, which ultimately fold into shapes and become enzymes and functional proteins. So these molecules here, all of three of these play a role in taking the DNA and converting it into an amino acid chain that folds to make a protein. And if you have no idea what I'm talking about, you might have skipped chapter 7 and 8 over metabolism and growth. It may not hurt to go back to that, because I'll be assuming that you saw that, and I'll be assuming that you understand that. And so when I talk about this, I'll assume that you remember that all. Okay, so let's go ahead and let me get the review questions up here, and then we'll pause, let you look at those. Give me 1 second here. Okay, so here are the review questions for part 2. Go ahead and pause this, take a look, and we'll go on to the next part. Okay, so in this next part, we're going to take a look at what's called applications of the code, and this is condensed. So we're looking at just a brief part of what's in the text. So applications is in reference to how, at least in a big part, how DNA actually works in a cell, what it's actually doing. So we'll go back to the central dogma here in a second, but there's really two key parts of how DNA works, and they're called transcription and they're called translation. Transcription and translation. Transcription involves taking the DNA and converting it into the RNA, which is the first part of the central dogma. Translation involves taking this RNA, which is actually a type of RNA called mRNA, and then turning that into protein. And that's actually where the ribosomes come in. They're going to actually play a role in transcription. So use the central dogma to describe how transcription and translation occur. So this is going to be pretty easy. We're going to once again show the central dogma. And you'll know this so well by the time the class is over. DNA, RNA to protein. So transcription is DNA to RNA. That is what we call transcription. Converting RNA into protein is what we call translation. So really, the two key parts of the central dogma can be described by these terms here. Now, there's a lot of details. Again, one more detail here just to clarify. It's actually a type of mRNA in this process called messenger RNA, mRNA. This is an example of where you'd have a gene that codes for an RNA gene. And the RNA gene, I'm sorry, I was misspeaking there. Never mind that, excuse me. The RNA gene would actually be encoded for another type called transfer RNA. I mentioned that one earlier. We'll talk about that here in a second. Anyway, that's the idea of transcription and translation. That's how they fit into the central dogma. So when we're looking at this, we're basically talking about how we get from the DNA to the protein. And the way to get there is we go from transcription, and that carries over to translation. So there's a process for transcription, and then there's a separate process for translation. So some of the major participants in this, one of them is called an enzyme called RNA polymerase. RNA polymerase is an enzyme that will bind to the DNA, and it will actually do the converting where it basically uses complementary base pairing to convert DNA into RNA. And I've got a video I'm going to show you here in just a second that does that. So just remember that you have complementary base pairing with DNA and RNA. So what it's basically going to do is going to take the code, the nucleotide sequence. So wherever we see ATCG, it's going to take and add the complementary nucleotide to that. So we'd go from ATCG to AUCG. So RNA polymerase is going to do that. Video make that clear. RNA polymerase creates messenger RNA, and then messenger RNA plays a role along with transfer RNA during translation. So let me show you the video, and that'll hopefully make sense. So for now, you just need to know that these are some of the major participants, and I'm limiting the number here. Another one that you actually should probably go ahead and add, and we'll talk about it, and I'll include it, but why don't you just go ahead and add it now, and that is the ribosomes. You can say either ribosome or ribosomes plural. There's lots of them. They all do the same thing, but that's another big part of it. And ribosomal RNA, if you remember, makes up the ribosome, so we won't need to detail that here, but RNA polymerase, messenger RNA, transfer RNA, and then ribosomes are all really some of the big participants here. Now, we'll go back here just real quick. Transfer RNA is an example of an RNA gene, and it actually becomes this weird three-dimensional looking thing. This is actually an example of where RNA can actually work a lot like a protein, where it has a shape. So here we have a sequence of nucleotides, and you can see that here in an RNA form, A, C, G, C, U, U, A, etc., etc. And what ends up happening is that this RNA molecule is folded in a way that actually allows it to complementary base pair with itself in certain areas. So it actually folds back on itself. So it starts out as a big, long, straight strand. So imagine if you just separated these bonds and just straighten that whole thing out. It'd just be a big flat piece. But it's allowed to fold back on itself, and it can actually complement in a way that creates this weird, creates this weird three-dimensional, they call it a hairpin shape, or a hairpin loop is what it creates. So we'll come back and talk about these here in a minute. But that's an example of an RNA gene. It's a gene that simply makes an RNA molecule, and then the RNA molecule actually forms this three-dimensional structure that does work in a similar way that a protein does. So that's an example there. Okay, so let me just go ahead and say this next, getting into the details of this, it's pretty difficult to actually visualize. This is where the videos come in, but the process is very confusing. It's not intuitive at all. So what you have to do is just simply sit back and look at how it works, and not try and struggle with why it works that way. It's one of those things about life. Life is very mysterious, and the way it works is very, very odd. It works, it gets it done, but it's not something that you would draw up. It's not something you wouldn't envision it this way. So it's very, very counterintuitive in that sense. So you need to kind of think about it from an abstract point of view. Okay, real quick, before I show you the video, let me just walk through the book. This is some of the textbook images here, and they're not very good, but it does give you some visualization to start with here. So the first part, transcription, DNA into RNA, and we're talking specifically into messenger RNA. It begins with the segment of the DNA where we have our gene to how to make a protein. So this isn't a structural gene, and it's a part of the DNA that codes for protein. What's going to happen is wherever that gene starts, the DNA is actually going to unzip rather. It's going to split, and the hydrogen bonds are going to separate, and that's going to allow these two DNA strands to open up, is the way to describe it. The DNA becomes open here. Next, the enzyme RNA polymerase, shown here as this big circle thing, it's actually going to bind to a key starting location wherever the gene begins, and it's going to start to convert that into messenger RNA. Now, if you remember, I said that only one side of the DNA contains the genetic information. And so here we're looking at one side called the coding strand, the other one called the template strand. So information is found on only one side of this here. So what's actually going to do is this enzyme is actually going to line up here, and it's going to actually use the template strand here to create an extra copy. And why they call it that is because while this is it's the template strand that's actually being used, ultimately the information in the mRNA matches the coding strand here. So the coding strand actually contains the raw message. The template strand acts as the complement to the coding strand. So when you make a copy here, you're actually going to make the copy in the same form as the coding strand because of the complementary base pairing. So anyway, the idea is that the RNA binds to the template strand and starts to add complementary base pairs to it, and we get this RNA mRNA molecule that literally starts to form right off of the template strand here. Now this enzyme will move down the DNA, and it will unzip and then rezip as it goes, so it kind of opens and closes around the RNA polymerase. And as it goes, the mRNA gets longer and longer and longer and longer. Now, in reality, these things can be really, really long, and because the genes in the DNA are very long themselves. So this can go on and on and on. But when it's finished, the mRNA will physically separate from the DNA, and the RNA polymerase will dissociate and separate. And then this big, long piece of RNA will now just be floating separately in the nucleus where all this is taking place. And now you have a piece of information that can go on to be used to make protein, and that's going to happen at the ribosome. Going back here to point out what's happening here, in the cell, you have all the raw building blocks already available. That comes back to nutrition, and there's a lot of things that are going on behind the surface here. But the cell is able to stockpile raw nucleotides, the A's, T's, G's, C's, and U's that it needs. And those are basically just in a reservoir within the cell, so it can draw from those as it goes. And so ultimately it's creating this great big long mRNA doing this process here. Okay. The next part is called translation. Translation is definitely the more mysterious part of it. With translation, what's going to happen is you have these molecules called transfer RNAs, and those were shown here. Now, transfer RNAs have this unique structure in that they form in a way that creates two key units. One's called the anticodon, or two key regions rather. One's called the anticodon region, the other's called the amino acid attachment site. Okay? Now, what's going to happen is that the transfer RNA is going to move into the ribosome in a way that allows it to interact with the messenger RNA. So the messenger RNA actually is free floating, and it's going to then bind to a ribosome, and the ribosome is literally going to feed it through the bottom part of the ribosome here. And as it's fed through here, it's going to take that information and it's going to match it up with the anticodon region of a transfer RNA. Okay. Now none of this is going to make any sense right now, and that's okay. I'm going to show you the video, and then we're going to go over this more. So we're going to go through this in layers. So if it doesn't make any sense, which it won't, that's fine. Unless you've had this before so far, it's not going to make any sense, and that's okay. So the mRNA has got information, and it's going to match up that information with the transfer RNA. Okay. Now transfer RNAs, they actually carry amino acids. Remember, we have 20 amino acids, and remember that amino acids make protein. Proteins are a big long chain of amino acids. So the way you make a protein is by linking amino acids together one by one. And you start with one amino acid, and then you add an amino acid to that, and then you go from there. That's basically what's happening at the ribosome. What's happening is the transfer RNAs, they carry the amino acids, and what they do is they bind to the ribosome, and then they start to link their amino acids together. And as they link them together like this, they start to form a small chain of amino acids. These chains of amino acids get bigger and bigger as more transfer RNAs come into the ribosome and add to them. So here you see, going back, we see a ribosome starting with two transfer RNAs. One of them transfers the amino acid, or I said that wrong, two transfer RNAs with two amino acids. One of them transfers the first amino acid to the second. We get a basic double amino acid here. And then what happens is now the first transfer RNA exits, and we now have a single transfer RNA with two amino acids on it. This one will now move over to this other site. Another transfer RNA will come in, and now we'll link the two amino acids from the first part to this new amino acid here, and now we have three amino acids. Okay, this next transfer RNA kicks out, so it would have been this one here. So as this was the second one that came in, okay, it held the 2 amino acids. Those 2 amino acids are transferred to this new one here, and it now gets all 3. This one loses the 2, and it's ejected. And I'm not going to test you on the details. That part I don't really need you to know. Just basically what's happening is each transfer RNA is bringing in an amino acid, and the amino acid chain starts to grow with each of these new transfer RNAs coming in. If you study this real closely, you can see the logic and the pattern where we go back to transfer RNA 1 and 2. 1 forms a bond with 2, 1 ejects, 2 is left with the bond, 2 moves over to the second site, amino transfer RNA 3 comes in, 2 transfers its bonds to 3, 3 now carries the bonds, 2 ejects, 4 comes in, etc. Again, I'm not going to test on that, but as you go back and study it, the details are there. So ultimately what's happening is we've got a message in the mRNA, and that message is going to align with the transfer RNA. And amazingly, what ends up happening is that the amino acids that are being used here actually match the information here in the messenger RNA. So the messenger RNA contains the genetic code, which then aligns these transfer RNAs properly so that we get the right amino acids to make the protein as the instructions dictate it. So to clarify, really what the DNA is saying, what a structural gene, to clarify, is saying, is that we have a sequence of amino acids. So the DNA is essentially spelling out a sequence of amino acids. That sequence in reference to how many, what types, and what order. And if you remember back from chapter 7 and 8, that's really what makes each protein unique and behave differently and have a different shape and function, is what amino acids, what types, and what order. Those create different shapes, and that creates different functions of proteins. So DNA sequence spells out amino acid sequence, which then dictates different proteins. So when we go back here and look at that one more time, we see the sequence of the mRNA with a message that spells out how to make the protein, what amino acids to put into that growing chain. Okay, so let's watch that video real quick, and then we'll come back and we'll talk about a little bit more of the coding and make sense of exactly how this works in more detail. So here's the video.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, so as you can see from the video, it gives you a visual of how transcription would look and then how translation would look. Now, translation is especially difficult. It's very mysterious. The transfer RNA molecules seem to just glide in and out of the picture and seem to magically find their spot, and it's very hard to see how that really works. And I've been studying it for 10 years and don't fully understand it, and I'm not sure if anyone in the scientific community fully understands exactly how it works. We know it works, and we can see it work, and you can see that it works consistently, so it definitely works the way they say it does. I've never really seen a full explanation for every little detail for exactly how transfer RNAs move in and out and make their match and that kind of thing. So let's talk about what we do know. Going back, there's a connection between what we have in the DNA and then how that translates to the amino acids that make up the protein. And there's a language, as I mentioned, and the language, as I said, starts out with four nucleotides, A, T, C, G. T becomes U, but they translate the same. So 4 nucleotides. And that language then essentially tells the cell how to make amino acids. And the way an amino acid is encoded is in a form of 3 nucleotides called a triplet. So, for example, let's go back here real quick. An example is if we have like a simple DNA sequence of AAA. AAA is 3 nucleotides, and that can lead to the production of a single amino acid. This, I believe, would be the amino acid phenylalanine. I'm not sure if that's correct. But one of your 20 amino acids can be encoded by that particular sequence. So in the DNA, anywhere we see this, we get a message that says we need that particular amino acid. So the idea then is that the message of the DNA from a structural gene standpoint, remember that structural genes are what make proteins. To clarify, there's actually a different language when it comes to regulatory genes. So when we're talking about this, to clarify, we are talking about structural genes, and it says that right up here actually. So there's actually a different way that it works in regulatory genes. So in this portion, the structural genes, we have these sections called triplets. Those are the 3 nucleotides, and each different set of 3 can code for a different amino acid. Now, it turns out, if you do the math, that when you have 4 nucleotides and you have 3 positions to arrange them in, you have what is 4 to the 3rd number of ways to arrange them. 4 bases, 3 positions. So we only have 3 spots per amino acid. So if we take 4 to the 3rd power, which is 4 times 4 times 4, we get 64. And so with 4 nucleotides and only 3 ways to arrange them, we actually have a limited number of possibilities, which is 64. However, if you remember, we have only 20 amino acids. So we actually have more than the number of possibilities than we have amino acids. What this actually allows is for what's referred to as redundancy, which means that we actually have multiple triplet codes, 3x triplet code, per amino acid. So let's take a look at this figure here to better understand that. Also, let me say one more thing here. So essentially we can get amino acids from more than one triplet code is basically what that's saying. The other thing about it is that we have what are referred to as stop sequences. So we actually have a stop triplet. several of those. So we actually have directions for how to make a protein in terms of amino acids, and then we also have a spot on there that says, okay, stop here, and this is the end of that sequence. So that actually takes up some of those 64 spots there. So let's take a look here. So here is a figure out of the text, figure 9.13. And what it's showing is all these sequences here that make up each different amino acid. So off to the right here, we have our amino acids, 1, 2, 3, count them all up, and there's 20 there. And then we also have our stop regions, as I mentioned, stop sequences. And then we also have what's called a start sequence. But the start sequence also codes for one of the 20 amino acids. So it actually has a dual role. So it's still an amino acid. It's also, there's also kind of a designated start sequence there. So from this, you can also see the redundancy that I mentioned there. So you have, for example, with phenylalanine, you have actually 2 triplet codes, and with leucine, you have actually 6. These 4 here plus these 2 here. Isoleucine, 2 more, methionine, 2, valine, 4, so etc. So some of these actually have 4 or 2 or even 6 in some cases. So then you have the designated stop codons, stop sequences. Okay, so redundancy is in the fact that proline, for example, has 4 sequences that all code for proline. So we have multiple ways of getting the same thing. And then we also have extra sequences built into that as part of our overall sequence. So let's go back here just real quick. And the idea is that in the DNA, we have these segments of 3 units called triplets. Each one codes for amino acids. Now, the way the terminology works is that they say that when DNA is converted to RNA, we call those codons. So a codon is basically the RNA form of the triplet, which I know is confusing, but they're basically, most of the time they're just called codons, to be honest with you. It's kind of a semantics. It doesn't really make a huge difference, but you need to recognize that the term codon is really the most commonly used term and is referencing the form in the RNA. So if you look here, for example, this says figure 9.13 codons, and it shows them in an RNA form because you would see uracils instead of thymines. So I actually was wrong about phenylalanine. But anyway, I don't care if you know these. I don't care if you memorize the codes. The exact memorization of this is not important. Just that you understand that these are the triplets or the codons, and each one codes for a different amino acid. So when you start to look now, going back all the way to the structural gene and how you have thousands of individual nucleotides that make up a gene, what you're really seeing in a big part of that are little units, little segments of 3s. And each one of those then codes for a little piece of a protein. And this is really what transcription is all about. Is really, I mean, both, excuse me, but it starts in transcription with the coding here. And ultimately what we're doing is just copying the DNA into an RNA form. And what you should take away from the video is that when the RNA is created, it now becomes mobile. And let me just clarify that because it might help understand things a little better here. This isn't the full reason why this happens, but to clarify here, when you have a cell, here's the cell, and then here's the nucleus, and here you have the DNA. Really? Doesn't like that. Here's the DNA. This is where transcription takes place. Now, translation is going to take place outside of the cell. So this is ultimately then where the RNA is going to take place. So transcription, the conversion of DNA to RNA, is going to take place inside the nucleus. Once the RNA is created, recall that we have now this mobile piece of genetic material. This can now travel out of the DNA and into the rest of the cell. Why would it do that? Because this is where the ribosomes are located. See how I've set that up there? Ribosomes are outside the cell, or outside the nucleus. So the RNA is going to be created here in the nucleus where the DNA is, but the DNA is more or less trapped here, so it can't exit. So it creates this RNA mobile copy, and it now floats out of the nucleus and then can become translated by the ribosomes. And this is also then where transfer RNA comes into the picture. So being mobile like that allows this to take place in different locations. I like to think of the transcription as like making a copy of a page in a book. You can think of the book, go back to the DNA analogy, being a book. Transcription is like taking just a piece of the information. You don't want the whole book, but you want to take a piece of that information with you because you can't take the book with you. So you make a copy of just the information you need, and now it's mobile, and you can take that with you. That's kind of the way I think about it. So that's where you can then see a little bit more on this. So when you go back and you watch this again, or you look at the video again, and we see, going back to this figure, for example, we have sequences here in the mRNA in codons. Now let's go back one more and let's take a look one more time at the transfer RNA. And remember here that we have what I called earlier is the anticodon region. And really I don't think I said much about it, so let me clarify that now. This is called the anticodon region, and this is a spot on this transfer RNA that has a very specific sequence, and this is always going to be complementary to one of the codon sequences in the mRNA. Now what's amazing about this, and this is just one of those, to me, kind of a breathtaking feature of life, and I know it's maybe hard for a lot of you guys to grasp all this, but one of the most amazing things about this is that the way it has to work is that the transfer RNA has to match somehow to the mRNA here. So remember I showed you that earlier, but I didn't really say how or why. So the mRNA, the mobile message of the DNA, carries codons, and they give the information for how to make proteins one amino acid at a time. So how do you actually make the protein? Remember, that's where transfer RNA comes in. It carries the amino acids, and they have to then match this sequence. So in order to get the protein to actually look like what the message says it's supposed to look like, you actually have to have a way then that the transfer RNAs bring the right amino acid at the right time and match this sequence. It's actually a pretty amazing thing, because that's exactly what happens. So what ends up happening is we always have the same start codon, and that, if we go back here to the list, there's always kind of a designated start location, and that's assumably kind of how Mother Nature always starts the process, with a single unit designated as kind of the beginning point. So you'll always find that at the start of a gene. It's kind of a way scientists can find the starting location of a gene, kind of. So there's always that start location there. After that, though, there's any of these other amino acids that are possible, minus the stop codons. Those, if you haven't guessed, are going to come all the way at the very end and tell the protein when to stop, or when the amino acids should stop being used. In other words, when the protein stops being produced. But we have a start location here. Now, any of these other amino acids, or codon sequences, can occur after that. Now, what's going to have to happen is, in order for this to work, whatever codon sequence comes next, the cell has to provide a transfer RNA with an amino acid that matches that, so that it will allow it to bind and then donate that amino acid to the chain. So what essentially is happening, going back to the beginning here, what essentially is going to have to happen is that the cell is going to have to bring in transfer RNAs, of which, by the way, these are located in the cell. The cell creates a lot of these without any real prompting. It just does this automatically. So cells create lots of transfer RNA as part of their everyday activities, and these transfer RNAs then get charged up with the amino acids from incoming sources of nutrients, and this is why you need protein. So your body eats protein, breaks down to amino acids, then your body uses those amino acids to build its own proteins. So essentially it stockpiles amino acids in the cytoplasm. So you have all these amino acids from your diet. Those then are being charged, or added to rather, these transfer RNAs that your body is creating, and then they're all floating around in the cell, and they get sucked into these ribosomes, and they're used to then create protein. Now, the way it has to work accurately then is that you have to have what's called an anticodon region of the transfer RNA. And the anticodon region is a 3-letter sequence, just like the codon, and it's going to be complementary to a codon sequence. And going back to what I said earlier, I kind of lost my train of thought there. I apologize, but when I said one of the breathtaking parts about life is that you'll always have a codon sequence here that is complementary to an anticodon sequence in a transfer RNA, and the codon sequence here will always match the amino acid in the anticodon sequence of the transfer RNA. So let's use phenylalanine as an example, and I'll abbreviate that as PHE. So and we'll look at this code right here for UUU. So 3 uracils in a row is a code for the amino acid phenylalanine. So in our RNA, our messenger RNA, we would see the sequence UUU. What we would see from our transfer RNA, let me try and draw this here. The transfer RNA looks something like this. Leave that spot open there. And what we would see right here is an anticodon region of AAA. So wherever we have an A, we would complementarily match with a U. So these two would be complementary to each other. AAA and UUU would be complementary. So wherever we see this in the RNA, what has to happen is a matching transfer RNA has to come in, bind to that site, and when it does that, it's going to be carrying automatically the correct amino acid, phenylalanine, PHE. So Mother Nature has it set up so that the amino acid that matches the codon also correlates with the anticodon of the transfer RNA. So as the cell is building protein, it's using this entire construct to make that happen, matching anticodons with codons and then allowing the correct amino acid to take place. It's really, it almost seems impossible that this is the way it works. But science has been working on this for a long time. It's not to say they have everything worked out, not to say they couldn't figure out that maybe they were wrong about something, but this seems to be fairly well understood, and there's some mystery as to exactly how it works. But generally speaking, that this is pretty well documented in science. So one more time, the messenger RNA has got the code in the form of triplets. They're being fed through the ribosome, and the transfer RNAs are then complementarily trying to match up. And then essentially when they do match, that means that you have a match for that amino acid that goes along with it. Now, from my understanding, what can happen here is that many of these can get rejected. My understanding, and I could be wrong about this, to be honest, I'm not sure if this is accurate, but from what I understand is that the ribosome will actually bring in random transfer RNAs and can then reject them if they don't match. And I believe that's part of how that demystifies it a little bit. It doesn't always just magically get the right one. It can get the wrong one, and that can then lead to a rejection where you'd have to pull in another amino acid. It has an amazing amount of accuracy, and it seems that somehow it has a good idea of how to pick the right one to reduce the number of errors, but it's not always a perfect match. Now, there's another way that this works in a more efficient way, rather, and this actually goes back to the redundancy of the code. Because really the truth is you actually have 4 different nucleotides that all code for the same amino acid, and it also works that there's what's called wobble, and you don't have to write all this down. I'm not going to test you on it, but just to help you understand it a little better, wobble is a phenomenon by which if the sequence is close, then it will usually match. So if you're one nucleotide off, they're close enough that it will still get the correct protein, or the correct amino acid rather. So if you look back at this real quick, if you have, for example, UCU or UCC, they both code for serine. They're close enough that you get serine every time. So this last amino acid, the last nucleotide can vary, and that variance there can actually still get you the same amino acid. So that's a little bit more complex part of it, and I'm just gonna go ahead and stop that there so we don't get too much more confusion. But the idea is that it's very autonomous, and at the same time there's rules to the way it works, and it's kind of governed by a certain flow of these processes in the cell. So that's the very confusing topic of transcription and translation. The last part here is just to talk about the fact, to go back one more time and reiterate the fact that ultimately then the proteins create the phenotype. So the genotype is essentially outlining the sequence of codons and the specification of protein structure. Phenotype then is what results as a consequence of that protein. What is the function of the protein? How does that function create some kind of trait, or how does that dictate something that's happening in the cell? So the phenotype is ultimately the consequence of how protein works in the cell. And then in other cases, RNA can regulate as well. But phenotype usually is in relation to the way the proteins work. So we get phenotype really as a consequence of translation, and it's the genotype that ultimately dictates the beginning part, which is transcription. Okay, so let me pull up the review questions here and let you take a look at those. Okay, so here are the review questions for part 3. Go ahead and pause this, and we'll take a look at the next section here. And we're going to take a look